Welcome, everyone. Great to have you here with us today for our 71st episode of This is CDR. This is CDR is an online event series presented by OpenAir to explore the range of carbon removal solutions currently being researched, developed, and deployed, and to contextualize them for policy proposals OpenAir seeks to advance at every level of government here in the US, as well as in national and subnational jurisdictions globally. My name's Toby Bryce. I work with OpenAir on CDR policy and market development. If you haven't done so already, please introduce yourself in the chat and tell us where you're tuning in from, and make sure you direct that to everyone and not just hosts and panelists so everyone can see your message. Just some quick background on OpenAir if you're not familiar with us. We're a distributed volunteer network dedicated to advancing carbon removal. We're a global community. We work together on shared open source missions in the areas of policy, innovation, messaging, and activist market development. There's a link in the chat where you can join us and see what we're doing. And I'll share a few links in a moment relating to a couple of these other things. My colleague Megha is having a bit of technical trouble, I think, and so I might have to do this beginning part by myself. Two quick things, and I'll put these links in the chat as well. We have active bills in California and Massachusetts. So if you live in those states, we can really use your help. And I'll put a couple links in the chat about the bills and a tool that you can use to call your local representatives in each state. But these are two really important state level carbon removal policies that are currently in committee, about to receive a vote. And we could really use any sort of in-state advocates to help us support the bills. Just some quick background on defining our terms. As always, this is the definition of carbon removal from a resource called the CDR Primer, which is a great way to learn about carbon removal. It's put together by upwards of 50 CDR experts led by Jennifer Wilcox, who's currently the Assistant Secretary of the Department of Energy, leading the Fossil Energy and Carbon Management Department. It's essentially the same definition that the IPCC uses as well, so it's really a great basis for talking about carbon removal. The definition is purposeful human activity to remove CO2 from the atmosphere and durably store it in geological, terrestrial, or ocean reservoirs or in long-lived products. As always, two quick things that we need to talk about up front when we talk about carbon removal. Number one, carbon removal is in no way, shape, or form any sort of substitute for reducing emissions as quickly as possible and decarbonizing our economy. 90+% of our climate work is to reduce emissions, and the purpose of carbon removal is to balance out the emissions that are difficult to abate in a climate relevant time frame. These are primarily from agriculture, and this is on the order of 5 to 10% of global emissions, and that is the job of carbon removal. And it has no purpose if we don't reduce 90% plus of our emissions, so we need to keep that in mind. That said, carbon removal there's clear scientific consensus expressed very clearly in the most recent IPCC report that carbon removal will be required at gigaton scale by mid-century. That's billions of tons per year to meet our climate goals. And we are currently at kiloton scale, so we have quite a bit of work to do. The other key thing about carbon removal is to disambiguate it from quote unquote carbon capture, or CCS, which is capturing CO2 from an emissions point source, a fossil carbon emissions point source like a cement plant. That may or may not be a good climate solution depending on the techno-economic circumstance, but it's an emissions reduction and not carbon removal, which again is removing CO2 from the atmosphere and durably storing it. Moving on to today's show, we're very excited to have the CEO of Andes, Gonzalo Fuenzalida-Meriz, pardon me, and he's going to talk to us about the very exciting microbial mineralization of carbon dioxide that Andes approach enables. And just a couple quick show notes. Gonzalo is going to give a presentation for maybe about 20 minutes. We have a few prepared questions for him, and then we're going to open it up to audience Q&A. And please, for our sanity, please put your questions in the Q&A box. That makes it much easier for us to manage them because there's other things happening in the chat. So using the Q&A box for that would be fantastic. So yeah, Andes, he's going to present the company's innovative approach that leverages microorganisms to accelerate the sequestration of inorganic carbon in soils. And a quick bio of Gonzalo. He founded Andes in 2018, along with his co-founder, Tania Timmermann. Gonzalo is a serial entrepreneur with more than 15 years of experience successfully leading biotechnology companies, background in business administration and economics, and experience in investment banking before becoming an entrepreneur. Originally from Chile and is now based in the Bay Area in beautiful Alameda, California. Gonzalo, are you ready? We'd love to have you come on and tell us about yourself and Andes and what Andes does.
Thank you, Toby. Excited being here. Thank you for the invitation, and thank you for the introduction. So yeah, let me share my screen, and I'll start a presentation. Okay. All right. Can you see my can you see me changing slides, Toby?
Yeah, right now it says loading.
I don't know why that happens. Yeah, let me. I will. Okay. All right. Yeah. Let me put in a. Sometimes this happens. I apologize. All right. Yeah. Sorry for that. Okay. Let me start with the science of what we do, Toby. You made a brief introduction on a high level what we're doing, which was great. Basically, what we're doing is that we are working with microorganisms that have the ability to live with plants, very intimate on the root structure, and while doing so, they have these microbes, the ability to capture CO2 from the soil air and convert it into carbonate minerals. Here, I like this image just to visualize the interaction between our microbes and roots. What you see on dark green on that image is a plant root, specifically a corn root, and then we have one of our microbes that we engineered it in order to include a green fluorescent protein gene, which basically allowed us to see it in contrast on the microscope and to visually assess where our microbe is living. And here you can see how our microbes lives very close to the plant on a condition that is called mutualism. And mutualism basically means that both the microbe and the plant are benefiting from living together. The microbe, our microbes benefit because they get their energy from the exudates from the roots of the plants, and in exchange of that, the microbe have different tools and capabilities that provides benefits to the plant, and being some of those. For instance, more nutrient availability. Also, specifically, this microbe has the ability to induce the formation of inorganic carbon. This inorganic carbon that the bacteria can create, it is typically it takes forms of calcium carbonate. And I'll be speaking during this presentation indistinctively about calcium carbonate or calcite, which is basically the same. Also, while speaking about carbonate minerals, carbonate minerals can take other forms rather than calcium carbonate, but we typically speak about calcium carbonate because among the cations that mineralize, calcium is the most abundant on the areas where we are working. So in this slide, we are basically showing on the left our microbe growing in vitro in our laboratory, and there we assess the formation of these structures that are these minerals. On the right side of the slide, we show basically results from an experiment, and this was part of the first experiment that we were realizing, right? When we saw this precipitation or formation of calcium carbonate, of inorganic carbon, we wanted to understand where this carbon was coming from. And by these kind of experiments, we show that the carbon containing these minerals minerals that the bacteria is producing comes from the atmosphere. In this specific experiment, what we did is that we have a control environment where we have the atmosphere of this where the bacteria is growing enriched with carbon-13. So basically, after conducting the experiment, we collect the minerals that are created, and then we send those minerals for analysis, and then we find that these minerals are highly enriched on this carbon-13 that was originally on the experiment on the atmosphere. So this type of experiments we've been conducting for the last 3 to 4 years in order to characterize and understand well the mode of action, how this bacteria has the ability to interact with the plant, and while doing so, it has the ability to create this inorganic carbon in the soil. Other kinds of experiments that we typically conduct are plant assays on a greenhouse environment. Here as well, we are looking for the buildup of this inorganic carbon on the soils. When we talk about inorganic carbon on soils, the way that we measure it is through what is called CCE, and CCE is an acronym that stands for calcium carbonate equivalent. Basically, there are different ways to run this measurement, but basically what we're measuring is we want to understand all the carbon that is contained on inorganic forms in the soil, and which after the processing of the soils, that form typically takes place as a carbonate mineral. Other types of experiments that we conduct, and this is where our research and development team is most focused right now and moving forward, is a mesocosm studies. We have columns where we understand and we measure very well every single or most of the characteristics of that soil and the leachate, what percolates through that soil. And we have also plants present with the microbes with different conditions, and this enables us to be able to have a full mass balance analysis of everything that is happening regarding carbon on these different conditions. Calcium carbonate formation, it is tricky. Pedogenic calcite or calcium carbonate formation is a slow process, and basically what we are doing is that we are using this microorganism that enables this process of generation and or precipitation of calcium carbonate to happen rather quickly. During our experiments that we run on the laboratory, greenhouse, and also on the field trials, we can assess and measure a rapid buildup or creation of these calcium carbonate minerals by our bacteria. I like this specific figure, but it is kind of complex, but basically summarizes very well the process of generation of calcium carbonate. That line that is in red is where equilibrium is found. If you go to the right of that red line, you will see a supersaturation of calcite, which means that those are conditions under which you will see the formation of the mineral or the precipitation of the calcium carbonate mineral. If you are to the left of that red line, you will see under saturation of calcite, and on those conditions, you will observe basically dissolution of calcium carbonate. Here, basically, what we understand, and after a lot of studies of these microbes, is that the microbes enable both the increase of the pH at a very under microenvironment, and also they enable the availability of calcium in solution, which is required in order to observe this formation of calcium carbonate. These bacteria, for many reasons, have developed the ability to, while working on living with the plant on the root environment, to generate this microenvironment with high pH and with availability of calcium to induce the formation of these minerals that contributes to the overall health of the soil and the plant. And this is a question that we get asked quite often. Toby, the question is, are carbonates good in the soil? And the answer is yes. This is a figure from a paper that basically summarizes very well all the impact and conditions under a soil with low carbonates versus a soil with high carbonates. And I won't go through all these different pieces of what happens on either conditions, but typically with a high carbonate content, you will see a higher resistance to pathogen pressure. You will see a balance, an equilibrium on the combination of fungi and bacteria. You will see more nutrient availability, and you will see also a better aeration and water permeability. Again, all these characteristics of the soil contributes to the overall health of both the plant development and the soil stability and health. We've been conducting during the past 3 years field trials, mostly here in the United States, across thousands of acres. Our last program during season 2022, we basically deployed our microbe across a little bit over 25,000 acres. These were roughly 200 fields scattered across North Dakota, Minnesota, and Wisconsin mostly. The kind of crops that farmers were growing there were mostly soybean and corn, but we also started trying with and assessing the performance of the microbe with wheat and sunflower. Roughly 10% of the fields where we worked last season were controls, untreated controls, which basically set the baseline for our claims for carbon being removed. One thing that is common across all these fields is that typically, I mean, all of these fields have a relatively high cation exchange capacity that speaks of available cations, most importantly, calcium and magnesium for our purposes of mineralization and buildup of soil and organic carbon. Cations that are readily available to being interexchanged and thus used. Also, another characteristic that is very important across these fields is that all of these fields have what is called as a very high buffering capacity. Soil buffering capacity means that a soil will change little its pH after being influenced by acidifying or alkalinizing factors. And this is explained basically because of the composition of these soils. Typically, the soils will have a lot of primary minerals, a lot of specifically silicates, in the range of 15 to 40% when we measure those on the A horizon of the soil profile, or which is referred as a topsoil. And lastly, but not least, and connected with the latter, is that these soils have a relatively high pH. The average soil pH of the fields that we worked during last season was roughly 7.2, 7.3. All these characteristics of the soils are ideal for our pathway, our strategy of using this microbe for the purpose of increasing the soil inorganic carbon. The process of, and this is part of the challenge of what we're doing, Toby. What we're doing right now is that we're measuring the carbon being generated and the carbon being removed directly on the soil. That means that we need to do a lot of hard work towards being able to understand the characteristics of every single field and have a system and a process by which we can collect the samples and for that collection and that observation to be truth to what is happening actually on the soils. And fortunately, this is an old problem that soil scientists have been dealing with for a very, very long time. So it is well understood which are the procedures by which a company like Andes has to pursue in order to be able to have a good understanding and observation of what is actually happening on those fields. Results from last season are very encouraging. We observed a huge increase on the soil inorganic carbon on those fields which were treated with our microbe. We observe statistical significance on the buildup of soil inorganic carbon on these fields, and when we observe what happened on those control fields, we didn't saw that increase or statistical significant increase on the buildup of soil inorganic carbon. All the information regarding these field trials and also information regarding in more detail about the mode of action of the bacteria and specifically how the bacteria manage to acquire cations from different compounds in the soil will be included in a paper that we are submitting during the upcoming weeks. This will be a peer review paper that basically will showcase the results of our field experiments and also in vitro experiments, and also will explain and we'll talk about how the bacteria is able to do this process of soil inorganic buildup in soils. Looking into the MRMV, and I am a little bit over, so I will go quickly through these slides, Toby. We adapted this verification framework from work that Carbon Plant did for enhanced weathering pathway. This wasn't created by a Carbon Plant. Hopefully in the future we'll have something similar for our pathway. But basically here we show the verification framework, which is basically focused on the measurement of the microbial induced calcium carbonate precipitation, or in more simple words, the buildup of soil inorganic carbon by the presence of our microbe. In regards to our challenges as a company, as I mentioned before, the process of collecting samples, processing samples, and doing analysis is very intensive. And as we grow and as we plan towards getting into megaton and later gigaton scale, we are thinking and implementing technologies in order to streamline this process of soil sampling collection and analysis. On the soil sampling collection, we are planning for next as soon as next year, and also at the end of this year to start the process of automatizing the soil sampling on some fields. On the soil sampling processing, we are also planning on doing automation on pieces of that process. And lastly, but not least, on the analysis on the soil sample for determination of the amount of calcium carbonate equivalent percentage of that soil. We have already developed a high throughput method, which is based basically on the use of a gas chromatograph, which we can very easily and fast and precisely measure the amount of calcium of CO2 that is contained on the soil sample by carbonate minerals. When we think about building trust on what we're doing, Toby, a big part of our a big piece of the team at Andes has been focused on developing an ISO standard methodology. We've been developing this document with the help of eco-engineers and a lot of external parties that have been contributing. We're planning for this methodology to be publicly available during next month. We think that we are setting a very high bar in terms of requirements and rigorosity by which we do all the process of including in our pathway. As part of the process of external validation, we will have an external party that will be a validation and verification body, and we're planning hopefully to be able to issue our first verified credits by the end of this year, which will be a vintage from past season. In parallel, we, as I mentioned before, we are working on this peer review paper, and in parallel also we are studying collaboration with externals. Lastly, but not least, let me talk briefly about farmers, which we consider our partners. Here, basically, I'm showing how our commercial model works upstream. What we do, Toby, is that we have our microbes that we provide to farmers for free. These are free of charge. We don't make any claims on yield improvement to our farmers, our partners, although that we've been conducting field trials with these specific microbes for the last 3 years, and we've assessed positive yields on soybean, on corn, and on wheat. By participating on this program, farmers benefit by receiving an extra payment that is a guarantee at the end of the season. The addition of the microbes into their fields is rather easy and frictionless. The microbes comes in a liquid solution, is very stable even at room temperature. It is just mixed with nutrients or with chemicals that are used for coating seeds, which is very common for soybean and wheat, for instance, to put fungicides. And the farmer just have to put in combination with those chemicals that they already apply to their seeds. They just add into the mixture our liquid product. In exchange of this, we ask farmers for them to provide us access to their fields. As I mentioned before, an important part of our MRMV is based on actually retrieving soil samples and measure those for the direct quantification of soil inorganic carbon that we are building up on the soils. And that's it, Toby. That was a presentation.
Excellent. Thank you so much. That was really great. Appreciate all of the technical detail. So as always, we have a few prepared questions from Open Air to walk through, and then we're going to open it up to audience questions. And I think Megha will hop on and organize those. And again, please put those in the Zoom Q&A box because it's very difficult to manage both the chat and the Q&A. So just put your questions in the Q&A box and we'll get to as many as possible in a few minutes. Just to kick off, we always like to hear a little bit about where you came from personally. Would love to hear just when did you start thinking, when did you, from Chile, when did you start thinking about climate? When did you start becoming aware of the idea of carbon removal? And maybe just a quick origin story of how you met your co-founders and formed Andes would be, I think people would love to hear that if you don't mind sharing.
Yeah, I just reminded something. As you mentioned, Toby, I am originally from I was born and raised in Santiago, Chile. Santiago has been a city that has been having a contamination problem for a long time. It is on a valley that has a poor ventilation. I remember as a kid that was an issue, and I also at that point share a passion for nature. I always tell the story about how my sisters used to mock me and call me Mr. Nature because I could be staring into a tree for 30 minutes, just appreciating what's going on, right? So yeah. When we started Andes with Tania, I met Tania through her PI. At that point, Tania was working as an investigator on a university. She was studying interaction of microbes and plants. And Bernardo Gonzalez, which is a renowned environmental microbiologist and a good friend, told talk me about this idea of how microbes could shape agricultural processes towards making them more sustainable, and that intrigued me and called my attention, and basically one thing took to another, and we started this. Now, at that point, Toby, the focus of the company was very different. At that point, Bernardo and Tania specifically had a lot of experience working on with PGPRs, plant growth, plant growth promoting rhizobacteria, basically microbes that have the ability to for different through different mechanisms increase the yield potential of plants. So we started working with different vegetables. I remember we did a lot of work with lettuce and with tomatoes, and one thing took to the other. At the end, Toby, we end up moving here to the United States, looking for great partners and investors. And yeah, for obvious reasons, then we changed our focus into row crops, right? Because that's basically yeah. So yeah. And the program, the carbon program, started about 4 years ago. We were looking for the ability of some of our microbes, the microbes on our collection, to be able to, one way or the other, to increase organic carbon in the soil, and yeah, we end up discovering this microbe that fascinated us, and its specific contribution was on inorganic carbon buildup on soils, and that is where we focus the program and the company then. Yeah.
Very cool. Great to hear. And just how did I find it really great that you guys are based in Alameda, which is, there are not that many startups based in Alameda. How did you end up in Alameda? Alameda is a very cool little neighborhood near Oakland, or in Oakland on the East Bay.
Yeah, it's beautiful and it's very tranquil, and we have a lot of sunny days. Yeah, we ended up there, Toby, basically because there wasn't much space in Emeryville. So at that point, so yeah, it was, we didn't have many options. We started on a warehouse on Emeryville, where we worked for roughly 2.5 years, and then we have the opportunity to move to Alameda. Yeah, we love Alameda. I mean, it's a beautiful, and yeah. We feel grateful at being able to be there. Yeah.
Cool. I will move on to slightly more technical questions. So when I first learned about or started reading about Andes, I had this question about the additionality of what you're doing, and you spoke to this in your presentation, but I'm just going to recap what my question was and what I think the answer is, and would love to hear you tell me if that's correct or wrong or what.
Sure.
So when I looked at the process, you're forming carbonates, your microbes are accelerating, I might not be using the right terminology, but the carbonation of magnesium and calcium ions that are already in the soil. And so they're taking up or absorbing these ions, turning them into carbonates, and that is leading to the carbon removal. And my question was, if you're not adding new calcium or magnesium ions or alkalinity to the soil, is that additional? Are you just accelerating something that would have already happened? And I think the answer is that you are working with soils that already have an extremely high concentration of calcium and magnesium ions. So there is additionality.
Yes.
Is that correct?
That is correct, Toby. And also it's important to mention that the pedogenic calcite formation, it is slow. It takes decades or centuries or even thousands of years to observe this buildup of soil inorganic carbon in carbonates forms. So we are basically accelerating. And the reason why it's so slow is because soils are in equilibrium, right? And what our bacteria does is that it has the ability to increase the soil pH in its microenvironment, and also increase the availability of these cations in solution, which is both required in order to see the precipitation and formation of calcium carbonate. So yeah, the additionality is brought by the presence of this microbe working towards creating this inorganic carbon in the soil. Yeah.
Okay, so you're accelerating, the process would have happened, but at more geological or century or millennial scale, you're making it happen much more quickly, years.
Yeah, and that would be true also, Toby, without anthropogenic intervention, right? When we think about agriculture, we are basically adding every year acidity into the system, which is the quantity that you need in order for observing carbonate formation. So yeah. And under conditions without humans, these processes happens very slow. With humans present and performing agriculture, you might expect the contrary to occur. So there is additionality.
And sorry,
Yeah, go ahead, Toby.
go ahead.
No, yeah, no, another important aspect, and this has to do with our methodology. For us, it's very important to have control sites. These are sites that share the same crops grown with the same practices, and soils that are very close by, or even patches within the field that share the same characteristics of the soils. And that is our baseline from which we start and compare the buildup of soil and organic carbon that we are creating. So yeah. That is another way of looking into additionality.
And just what you mentioned, acidification or pH, and so if you're absorbing these cations, you are reducing the pH. But is it because you're already working in high pH soils that this is not a problem, or what is the effect of your process on potential soil acidification?
Yeah, that is a good question, and the answer is right. It's because these soils already not only have a high pH, Toby, but have a high buffering capacity. All these primary minerals that are present on the soil consumes acidity. That's the reason why you won't see a pH either going up or down. There's an equilibrium on these soils. So it is very different if we were speaking of soils with a relatively low pH and low cation availability, right? On those conditions, what you are mentioning that would be true. You will observe a depletion of these cations and further acidification of the soil, which is not the case.
Okay.
In our
and just one last question on this topic. It sounds like a great place to deploy if you have large surplus of cations and high pH. Is that a common soil type? Is that like, or have you done an analysis of global?
Yeah.
And like how much of the global geography, land-based terrestrial geography, is suitable for your approach?
Unfortunately, there's not enough information to claim anything on a global scale, Toby. But what we've done is that we've mapped the Americas, North America, including Canada and the United States alone. Those are there is roughly 200 million acres of agricultural land that has the ideal conditions for implementing this technology. If we look into South America, there are other roughly 200 million acres, most importantly in Argentina and Uruguay, which have soils that have the right characteristics for implementing this technology. We will be looking into other areas. Andrew Leno from our team has a love for Africa and Kenya. So yeah, there are many places, Toby, where there are soils with a lot of cations and a high pH, but they lack water. So that is a factor that we look for. We need water in order for these processes to happen, and we also need agriculture to be already happening. We wouldn't love the idea of transforming land into agricultural land for the purpose of just so we can leverage on the existing capacity, and there's plenty already. Yeah.
I'm going to leave any follow-ups to the soil scientists and geochemists in the audience, because we've definitely reached the extent of my knowledge here. And also a big shout out to Andrew Denu, who is fantastic and helped organize this session. So hello, Andrew. Can I ask one more question? You mentioned moisture, so you need moisture. Is temperature a factor? Obviously, you're deploying in the great upper Midwest, where I guess it's cold in the winter, but not in the summer. Like, is temperature a factor as well?
Pedogenic precipitation of calcite requires temperature for that to happen. In our case, it's a microbe that is leading. This is a biogenic approach, right? We work on soils which have lower temperatures compared with the ice states, for example. We're focused on northern states. We haven't seen a detrimental performance of the microbe in regards of building up soil organic carbon on those kind of soils. So I would say that you need a temperature, but not extremely high temperatures. Typically, planting in northern states starts with a soil temperature of 10 Celsius degrees, 10 degrees Celsius. So yeah, we don't need high temperatures for this process to happen. It's not a requirement.
Two more quick questions, and then we do have a large number of audience questions. And today is a crazy day on this is CDR. My dog is going crazy. So number one, is it possible? See, I believe your primary delivery mechanism is via coating the seed with the microbe. Is it possible? Is it possible to work with other soil elements, whether it's fertilizer or what have you, or are you restricted to deploying with the seed?
No, it's not restricted. Actually, we have farmers during last season and during this season that are applying the microbe in combination with fertilizer. There are others that also have in furrow application equipment. It is equipment that puts the seed and also an inoculum of the bacteria at the same time. And there are others that combine our microbe, our liquid product with the typical chemistries that they apply as a coating on their seed. And that is very common for soybean and wheat, Toby. You won't see seed typically comes naked, seed for soybean or wheat to the farmer or the distributor. So yeah, it is very compatible with many ways of application.
And is it compatible? We had our friend Adam Wolf from ION on the show last week, who gave a really great presentation. Is your approach complementary to the application of silicate minerals like basalt or olivine? And would there be an attribution problem as to who gets the credit for whatever carbon removal is happening? What are your thoughts on that?
That is a good question, and I think that the way to answer is with empirical data. We don't have, unfortunately, that data on combining the application of olivine, for instance, with our microbes. But yeah, that would be. I don't know the answer for that, Toby. I would love to have a conversation with Adam and to explore that. But yeah.
Cool. I'll put you in touch. One last quick question. You had a great slide on how you work with farmers. And just to clarify, so is the farmer paying anything for Andes, and is the farmer receiving a share of the carbon credit revenue?
Yeah, they are not paying for anything, and they are receiving a share of the revenue. Yeah.
Cool.
Currently, what we are doing, Toby, is that we are paying a fixed amount of money by the end of the season, depending on how involved is a farmer. Yeah, and that has worked with us. We've had an enormous traction on the field from farmers. We're doing this season 50,000 acres. We have demand for over 200,000 acres, and we haven't spent a dime on communication towards farmers. This is just a word of mouth of farmers talking each other about the program and wanted to participate. So yeah, I guess what we are offering them is it's quite substantial. If you look into their numbers and how we impact their bottom line, is very impressive. Yeah.
Excellent. I have more questions, but I want to leave some time for, we have a bunch of audience questions. And if Megha is with us, maybe you can come on and package some of these up and ask Gonzalo. Cool. Hello, Megha. Thank you, Gonzalo. That was
Hello.
really appreciate you answering the questions.
Hi, Megha.
Hi, that's been really great. Sorry, everyone, for my technical problems earlier. Cool. So we had a bunch of questions. I guess I'll start on the technical side a little bit, and then there were some on commercial things as well. A couple of people asked about whether your microbes are similar to mycorrhizal fungi, and if you kind of see similar synergistic relationships with fungi, or like how is the similar or different to those kinds of things that people might be familiar with?
Yeah, that is a good question, and when we think about bacteria and different kind of bacteria, to make an analogy of how different they are is comparing a bird with a whale, a different species of bacteria, right? And the same is true with fungi, right? Now, what we know is that there's mutualism between fungi and microbes, and when we think about the addition of our microbe into the whole microbiome of the soil, basically what we are typically on a gram of soil, you will see roughly 10 to the 9 cells of microbes. We are taking a small percentage of that population. Typically, our microbe is in a concentration of 10 to the 5 per gram of soil. So what can I say is that our microbe is interacting with the whole community there on the soil. Now, understanding how the microbe shapes that community is more complex, and there are some studies that have found that microbes similar to ours increase the diversity, for instance, of the microbial community. But yeah, what I can say is that it is compatible. This microbe is a natural microbe that was collected from roots from a corn plant growing on soil, right? So yeah.
Okay, got it. And like, how long do these microbes that you add, how long do they persist in the soil, or is that something you're still kind of testing over time?
Yeah, we have a good idea that these microbes survive during the growing season. The image that I show at the beginning basically show how integrated this microbe is to the root structure of the plant. These microbes specifically depends a lot on plants. So after the growth season, the plant dies, and then a harsh winter comes into place. So yeah, we need to reapply the microbe after winter, because you won't see a huge population, or even being able to measure the microbe on the soils after the season. So yeah, they will stand for the season.
Okay, got it. We also got a couple of questions related to biochar. So I know Toby kind of mentioned the olivine as like a potential other additive. Does this work with soils that have added biochar? Is that something you're looking into at all?
We are not looking into that, but that is a good question, Megha. Yeah, I cannot think right now why it wouldn't work. Basically, these bacteria that we are working with gets most, if not all, of their nutrients from the exudates from the plant. So I wouldn't see a detrimental effect on the addition of biochar into the. So yeah, we're not testing that, but open to a potential collaboration on or testing that and assessing empirically if it's a good idea.
Okay, got it. And then in terms of the soil pH, so does the soil pH change much as you add these microbes, or are you changing the pH by adding other additives to sort of keep that in balance, or how does that change as the microbes interact?
Yeah, we're not adding anything in order to keep the soil pH in balance. The soils that we are working on are highly stable in pH. They have a high, what is called buffering capacity. That means that if you add a lot of acidity into that soils, that acidity will be consumed by primary minerals, mostly silicates that are present. So yeah, we don't see much variation after the season on the soil pH. Yeah.
Okay, got it. And then in terms of the plants that this work with, does this kind of capture capacity vary much depending on what kind of plant species are in those soils, or is it more dependent on the soil sort of quality itself?
That is an excellent question, Megan, and we haven't found the statistical significance on difference of buildup between, for example, soybean and corn, which is interesting. We would have expected a different, right? Because of different rates of respiration of different plants, right? Yeah. For now, we haven't seen much difference on the buildup capacity while working with different crops such as wheat, soybean, or corn. Yeah.
Okay, got it. And someone just had, I think, a clarifying question. So they're asking, are the microbes focused on just dissolving the calcium and magnesium into the ions, but not focusing on reacting the CO2 itself? Or like where exactly, they just wanted to know, I think, where exactly the microbes interact?
Yeah, that is a good question. Unfortunately, we have a good understanding of what's going on. I cannot go into many details, Megha, because we are filing IP.
Sure.
Now, information on that will for sure be able to answer that question will be contained on a peer review publication on the upcoming months. So yeah, bear with me on and that answer will come. But there are other elements present that are provided by the bacteria that basically it enables the capture of the CO2 and further availability of the cation and high pH in order to induce precipitation.
Got it. Okay, and I guess talking a little bit about the measurements around the carbon, we've got a question from Greg Rao, who's always a great contributor on these discussions. Have you been able to show that the total inorganic carbon storage, so that includes both dissolved and particulate matter, that number actually increases with your treatment? Or I know that there's like different ways of measuring these things. How do you look at that?
Yeah, the question, Megha, is that we've been able to measure the total inorganic carbon understood by the combination of soil inorganic carbon or the carbonates plus the dissolved inorganic carbon. That was the question. Mm-hmm. Yeah. We've done that on column on mesocosm studies. Doing that on the field is challenging for obvious reasons. Specifically on the fields that we are working, these fields have a high content of clay. So going very deep into the soils is always challenging. But yeah, on mesocosm work, and this will be contained on the paper that we will be publishing. We've assessed that we are increasing the total inorganic carbon, understood by the sum of the carbonate minerals plus the dissolved inorganic carbon.
Perfect. Okay. Just thinking a little bit about how big this can be. If you were to apply these types of microbes to all the kind of soils out there that you're aware of that this is potentially appropriate, how much carbon could this really address? And then a follow-on from that someone else asked is there a way to treat maybe more acidic soils or other environments to make them appropriate, or is that not something you'd want to do?
Yeah, good question. Thinking on and based on what we know right now, Megha, if we could apply these microbes on all the acres across the Americas that are suitable for this strategy, basically we're looking into being able to get into gigaton scale on a yearly basis.
Okay.
We need to better understand what's going on with the dissolved inorganic carbon on soils with a lower pH. It's not that the bacteria is not being able to contribute to remove CO2. It's just that we haven't been able to measure in fields. Part of the mesocosm studies that we are further developing is understanding this kind of phenotypes and results on different kinds of soils, including those with a low pH, which are plenty, right? If we have the ability in the future of including this lower soil pH into our program, that will be huge. Now, that will require some sort of modeling being present. It won't be only a direct measurement, but we will need modeling as other approaches do. But yeah, that is something that we're looking into.
Okay, great. I will just do two more before we go. So one person had a different idea for a use case. So saying, depending on what microorganisms you're using and what they're able to extract, would you be able to use this for something like cleaning up heavy metals from industrial contamination? And they mentioned, London, for example, where I live, being a lot of ex-industrial sites. Are there any other use cases like that are a bit more out there that you consider?
Yeah, I mean, yeah, and that is a good question. That field of study, Bernardo Gonzalez, one of the initial members of Andes and now part of the scientific advisory board, has focused most of his career on what is called bioremediation, the use of microbes in order to clean soils. And yes, it's something that could be done. Now that will require a lot of effort, investigation and money and time in order to, but yeah, it is something that I know several groups that are actually working on that. And we have that a big problem in Chile, from where I am originally from. There's a huge mining industry, and this is a huge problem in Chile, right? So yeah, I have some friends actually working on that back there.
Okay, very cool. Last question before we go. So just thinking about the next 5 years, what's the kind of pathway for the next 5 years? What kind of scale are you hoping to get to? And what are the key things that need to happen, policy, market, et cetera, to achieve that?
Well, market is important, right? Megha, we are looking into engaging with corporates that are thinking on the long term, that want to jump into long-term offtake agreements, that are looking for a scalable technology with a robust MRMP, and also with the right price point, right? And that's the thing that I guess everybody, we are all working towards, right? Being able to offer a carbon dioxide removal at a price point of less than $100. Our plans for the next 5 years, where do we see ourselves? We see ourselves at the megaton scale. We see ourselves being deployed internationally. I have a fascination for what we're doing, not only because of the impact that we're doing on the environment, but also the impact that we can do on people. I would love to see our technology well deployed into South America and impacting farmers and their communities there. And we see ourselves within the next 24 months being able to offer a carbon dioxide removal at a price point of below $100. So a lot of things will happen hopefully within the next 5 years.
Great, super exciting. Thank you so much for joining us. And Toby, I'm going to get this back to you.
Thank you, Megha.
Talk about the
Thank you to the audience for all the great questions. I'm sorry we didn't get to quite all of them, but we got to a bunch of them. And especially thank you to Gonzalo for being with us and for being such a good sport about answering questions and leaving on such a optimistic note for the future. Very excited
Absolutely.
for Andes and what you're doing next, and look forward to staying in touch. So thank you so much for being with us.
Thank you, everyone. Thank you, Toby and Megha. A pleasure.
I'm going to share my screen briefly, just a few closing notes. Number one, I mentioned I reposted in the chat the links to the California action and the Massachusetts action, both of which are really they're happening now, and we really could use help. Literally, calls can make a difference. So if you live in those states, please either connect with us or take down these links and call your state level representatives. Number two, we have a bunch of great upcoming CDRs. We have the folks from Frontier on next week to talk about their recently launched offtake purchases, and then a series of really exciting programs running through the end of the summer, with a couple more still to announce. So there is a link in the chat to the This is CDR page on Eventbrite where you can sign up for all of them. Would love to see you at as many as possible. And that's it for today. Thank you for being with us. Thank you again to Gonzalo for the great presentation, and everyone be well, and look forward to seeing you next week with the folks from Frontier.